Hi, everyone. Welcome to Chapter 2. We call this chapter Chemical Physiology. It sounds rather technical, and it can be technical, but we're not going to let it get too technical. We're going to try to introduce you to some concepts which will dovetail nicely with hopefully your current understanding of cell biology, but then begin to translate that into the certain types of clinical decisions that you may be involved with or may be managing as you manage a patient in a healthcare team. So let's move forward. Before we delve into the topics that we're going to delve into, and I'll give you the list of some of the things we're going to shoot for in this chapter, it wouldn't hurt you if chemistry is not your bag of chips, shall we say. It wouldn't hurt to go and review some of these concepts. Either look at your old notes or go in and look at ion transport a little bit. Well, that's from a lot of that's from 165 or basic chemistry, solutions, logarithms and pH. I'll try to break that out a little bit. Logarithm is a kind of a scary word for something that actually makes life a lot easier. It's just a type of annotation. We're going to talk about it when it comes to pH. If you remember acids, bases, and buffers, I do have some slides based on this in this presentation. So that one we'll actually talk about a little bit. And then the different biomolecules, it wouldn't hurt to refresh your understanding of those. We'll get into chapter 3 a little bit on this, but if you want to speed up the process, maybe review, especially Bio 165 if you've taken that course. I've often thought, why revisit things that we might have seen in 165 or chemistry or biochemistry and that sort of thing? First of all, it's because not everybody's had as much as we would hope for, especially your patients. I think we don't think enough about how we need to make ourselves as strong as possible for our patients. Our patients are the ultimate customer here. But those of you who are going into physical therapy and occupational therapy, those of you going into medicine, veterinary science, PA, dental, you name it, you're going to encounter, or as a patient, you are going to encounter pharmacology. All right. So this chapter deals with the basis of a lot of the things that we deal with when we're talking about pharmacology, which is the delivering of external components. That can be as simple as water or as complicated as, say, a hormone or something along those lines. That's pharmacology. So we need to understand the basis for that. Some of you have a good understanding already. We want to make sure everybody's on the same page. Nutrition. I don't think we spend enough time with nutrition either. I teach an FYS course where we try to get into the science of food, and food is so much fun. It's so delicious. It's so good for you. I like to say the 4 food groups are carbohydrates, proteins, nucleic acids, and lipids, rather than meat and dairy and that sort of a thing, because those are the 4 groups for biomolecules. So this is a key component of nutrition. Electrolyte balance, for instance, athletics, so forth. Then we have endocrinology, which is where we're going to go when we go into chapter 7. Endocrinology is basically pharmacology that is endogenous. That is, the body makes the molecule and the body responds to that molecule just like as if we had an exogenous molecule. Pharmacology, endocrinology is the same stuff, only it's done internally through chemical signals. And then lastly, and this one might hit home to many of you guys that are in athletic training or have an athletic background, inflammation. Inflammation is fundamentally sort of a combination of endocrinology. All right, it is an endocrine type process. Sometimes people won't say endocrine, they might say paracrine, but that's okay. It's the same stuff. It's just how far it goes in terms of distance it travels. But inflammation is the root of most, if not all, diseases. Either disease is an inappropriate expression of inflammation, or sometimes there are diseases where there is not enough inflammation. Either way, these are all connected by today's chapter, and I hope I can inspire you a little bit to try to scratch a little deeper than the superficial stuff. We're going to get you started today, and I hope you'll follow this through the rest of your careers because it leads to nothing but good things. On a conceptual level today, we are going to use the information that I'm going to provide you in order to help you understand more about non-covalent interactions, that is to say, the non-covalent bonds that give rise to things like protein structure, which then gives rise to protein function, which gives rise to cellular function and tissue function and so forth, as well as interactions. That is to say, how is it and then why is it that proteins do what they do? How is it that they bind together and dissociate or aggregate, et cetera, et cetera? So we're going to use the concepts today to get after those two major ideas. A quick laundry list for you for learning goals for this chapter, things that you're going to want to pay attention to, making sure that you've got a good understanding and functional ability are things like, for instance, ions. All right, so we're going to talk about ions all semester, especially sodium, especially potassium, but we're also going to talk about calcium and chloride, phosphate, all sorts of things. So make sure we get into understanding of what ions are, anions, cations, that sort of thing. Molecular bonds, to be able to recognize them visually is probably the most important thing for you guys to be able to do. Like, for instance, can you recognize a hydrogen bond? Why do we do that? Because the language of medicine, language of pharmacology and so forth is chemical. So be able to recognize a hydrogen bond when you see it shown on a piece of paper. Covalent bond, that should be another thing. And again, if you've had biochemistry, you've had organic chemistry, that should be old hat for you. So make sure you can recognize those in a structure and where they happen to pop up in the molecules that we're going to be talking about. We also have ionic bonds. We don't see these quite as often. You see these in proteins big time. It's a very, very important bond in what we call tertiary and quaternary structure. Tertiary is the folding of an individual peptide into a three-dimensional structure. Quaternary is the association of multiple peptides. So we see ionic bonds quite a bit in that sort of tertiary and quaternary situation, important for protein-protein interactions. And then we have what are called hydrophobic interactions. These are where instead of being a sort of a water-soluble association, it's a water-insoluble association. Many of your carrier proteins that carry molecules through your blood, like for instance hormones, have to have a hydrophobic center, or those hormones just can't do well in that aqueous environment. For instance, testosterone, estrogen, any of those stones, if you will, of lipid hormones need carrier proteins, and they're carrier proteins because they have a hydrophobic region. Understand the relationship between shape and function, proteins in particular. The shape is everything when it comes to function. Just like in anatomy, structure and function are deeply, deeply interconnected. Here, we're going to live in the world of the protein. pH, can't emphasize this enough, and this sort of ties us back to the very first point, ions. pH is huge. Why? Because it affects almost every level of structure when it comes to proteins. For instance, secondary structure, hydrogen bonds, pH. Tertiary structure has hydrogen bonds, pH. Quaternary structure, hydrogen bonds, pH. So it's huge. It's one of the key things in a chemical analysis is blood pH, and you die quick. One of the reasons we get hiccups when we laugh too hard is to break up the hyperventilation that you're doing when you're laughing that hard, which can lead to a huge chemical imbalance and cause you to pass out or even worse. Different functions of proteins in cells. This is where this all goes. Ions and chemical bonds help make the shape and structure of the proteins. pH helps ensure proper structure in the cellular and the internal environment of the body, and then that dovetails nicely into function. So we have soluble proteins. These are our enzymes, globulins, and so forth. And we also have insoluble proteins and understanding protein interactions. All right. We're not going to go too deep into this. We're not going to get into the complex mathematics, but we are going to try to understand affinity. We are going to define these things over time, especially in chapter 4. Okay. We are going to define why, because interactions between proteins and in each other and proteins and non-protein molecules are dynamic. They are not just on all the time, off all the time. They literally float on and off in a probability function, if you will. What's the likelihood that something's bound or not bound? That's what we call affinity. The higher the affinity, the more likely it is to reside in a bound form. Again, that's what we call the law of mass action. Getting a little further along here than I anticipated, but I wanted to make sure you at least understood why we're doing what we're doing here. So why dedicate the time to this stuff when we could be moving into more what feel like more relevant topics? And a lot of people come in to this class questioning, why do I have to learn this? Because this is where the action is. Everything that you do clinically should be rooted in we are biochemical organisms. So having some awareness of this will again tailor how we handle disorders and so forth. Things like why we have curly hair versus straight hair. We think a lot about neurons and the neuromuscular interface. Certainly muscular contraction is a big deal, but equally important is endocrine signaling. And we take advantage of that endocrine signaling using exogenous compounds, pharmaceuticals. Certainly understanding enzymes, a lot of disorders and a lot of problems in patients are enzymatic in nature. For instance, hemophilia, for instance, your blood doesn't clot properly. That's an enzymatic issue. More and more, we're understanding antibodies and immunity, whether we're talking about vaccinations or we're talking about therapies for cancer. There are more and more antibody-based drugs. I'd love to spend more time on immunology, and I think if you're going to go into healthcare, immunology is a really great topic to study. Genetics and so forth. A lot of stuff. All this stuff is based on the concepts we're going to talk about today, whether it's injury and pathophysiology or treatment, whether we're doing manipulations, putting things on patients, casting, training, pedagogy. All of medicine often involves modulating protein function in particular. If you've ever been involved in acute care or emergency care, or observed patients in emergency rooms, so forth, one of the things you'll often see, or if you're reviewing a patient's chart, you'll see these Chem 7, Chem 8 basic metabolic panels, if you will. So why are these considered to be so important? These ions and these functional molecules, why are they so important? Because that's how the cells get the job done, is using these different types of molecules. Things like ions and things like pH. So we want to talk about those things a little bit today, especially pH. We're gonna give some lip service to ions, but especially pH in this lecture. And so those ions and pH all help modulate protein shape. And if you look at this picture here, you can actually see a membrane here, and there's a cytoskeletal element here, and some kind of a globular protein, and so forth. This was a really, really amazing. It's an artist rendition of what looks like an organelle. There's an organelle here, and membranes, because all of these structures, well, even if they have, like, for instance, phospholipids, like in these membranes here, nevertheless, proteins are at the core of a lot of the function of these things. So we're going to delve into that topic today. So we have four major categories of bonds. We have covalent bonds, which involve the sharing of electrons to create a very, very, very strong bond. Mostly what you want to be able to do is recognize these solid lines between these atoms as a covalent bond. Maybe talk about that a little bit later when we get to disulfide bonds. We have ionic bonds, which are stabilized by having opposite charges on the side groups of amino acids, particularly in proteins. We have hydrogen bonds, which are not covalent, not ionic, but they have elements of both a little bit. Then we have the most nebulous, which is called the hydrophobic interactions, of which van der Waals is one of them. This is what gives oil and things like graphite its excellent lubrication properties, because these individual molecules here can slide across each other. It's the weakest of the forces. Starting with ionic bonds. Ionic bonds involve ions, if you will. That is to say, like for instance, an atom that has lost an electron, like for instance, a sodium atom, if it gives up an electron to say a chlorine, all right, that chlorine is going to hold on to that electron and not let sodium have any access to it. And so now we have a negative charge on the chlorine and a positive charge on the sodium, and that would give us sodium chloride. And that's an ionic bond because these two now ions are attracted to each other by virtue of their charge, the basis of their charge based on the ratio of electrons and protons. Opposite charges attract, identical charges push apart. And we'll get to that when we especially get into resting membrane potential. This has relevance in proteins. Protein amino acid side chains can, in aqueous environments, lose or gain charge. If they're acidic or basic in nature, they may give up a proton and thus have a negative charge, or maybe they accept a proton and have a positive charge, and that can create interactions that stabilize structure in proteins. And those are called ionic bonds in proteins. And that's why ionic concentration in intracellular fluid or in the blood, back to our chem panel picture, that's why ionic concentrations can be really, really important, because they have an effect on protein structure. And for delineation purposes, in terms of image, you should be able to recognize an ionic interaction in a protein. So here, it's a little hard to see here, but there's a plus and a minus here if you look closely. If you see that plus and minus, as opposed to say a dotted line or a solid line, this is a covalent bond here, a disulfide bond. This is a hydrogen bond right here. This plus minus, that's your indicator that you have an ionic bond, ionic interaction going on, either inside a given protein or associating with multiple proteins, or a ligand substrate type interaction. Above and beyond proteins, we do have very important ions in the body. And so you should be able to know what these and be able to recognize these. Sodium, potassium, calcium, hydrogen. These are all elemental ions. Chloride is in there as well. But you can also have multi-atom ions, like for instance bicarbonate, which we're going to talk about more later on in the semester. Phosphate, when we talk about protein modulation, is super important. Sulfates, I won't talk too much about those, but these are all really, really important ions above and beyond protein structure. Yes, they will influence protein structure when they're in extracellular fluid, but they also have individual functional roles as well that we're going to touch on moving through the semester. Now on to hydrogen bonds. Hydrogen bonds are a polar bond. They are a polar interaction, but they are neither covalent nor fully ionic, and they have very, very specific parameters. In biology, the hydrogen bonds we're talking about involve a bonded oxygen or nitrogen. There are other types of hydrogen bonds out there. They include fluorine and other things, if you get into inorganic chemistry. But in biology, then the vast, vast majority are going to involve either an oxygen or a nitrogen atom and a hydrogen. So here are two water molecules, and so here's that bonded oxygen, and it's slightly negative. That's what that delta negative means. A little delta negative means that there's a slight negative charge there. The electrons are hanging out with these oxygens a little more than they are with the hydrogens. That means the hydrogens, though, still have a little bit of, that means they have a little bit of positive charge, and so there's an interaction there. And this hydrogen bonding is what gives rise to these emergent properties of water that make water the molecule of life, if you will. We have trouble visualizing the possibilities of life without a molecule like water. The fact that ice floats and doesn't sink, the cohesive and adhesion, and so forth. But hydrogen bonds are super important in DNA structure. We have the double helix, if you will. One strand on one side, another strand on the other side is stabilized by hydrogen bonds. And you see these little dots here between these, either a nitrogen or an oxygen and a hydrogen. Super duper important in protein folding. All right. When we get to the different structures, secondary, tertiary, and quaternary structure, hydrogen bonds are going to be very, very important in those structures. And lastly, the van der Waals, I tend to just summarize that and say hydrophobic interactions. These are the weakest, and again, it's the force, or it gives rise to the properties of things like oils and non-aqueous lubricants, because these electrons are flowing in and around the molecules, and so while there is some adhesion, nevertheless it's a very, very loose association. So van der Waals is the most common, and we call these interactions hydrophobic. For instance, these fatty acids that are depicted in this membrane structure, this phospholipid bilayer, we have these polar head groups. These are more attracted to water and hydrogen bonds and so forth. But you have these non-polar fatty acids that are attracted to each other because the electrons can flow between them. That's that hydrophobic interaction. We're going to talk about this from time to time. Especially in chapter 5 with the membrane interactions and so forth. In protein structure, they add another layer of stability to a protein. So if we take a protein and we subject it to differing forces, if you will, what you see is you have this beautiful helix here that begins to disintegrate as we add force to it. Now, this is with van der Waals forces. Now, the experiment was done where they were able to substitute, and so you didn't have van der Waals forces. And what you see is the destruction of that beautiful structure is far more enhanced when you don't have those. So it's a subtle thing, but it's an important one. And when we get into hydrophobic interactions, when we're talking about especially membrane transport. I'm going to stop right here. We've gotten through the bonds, and we've spent a little bit of time here. I know that trying to soldier through all this can be challenging. So I'm going to take a break here, stop the video here. We will pick up Chapter 2, Part B in the next video. So thank you for your time. Please, please, please bring your questions. Take good physical notes. That really does help with the learning process. And I'll talk to you soon.